All righty, we'll get started. Thank you everyone for coming to the second hands online workshop that's organized by the Collaborative Microbial Metabolite Center. Um, these, uh, these workshops are recorded, and we will post them later on the Collaborative Microbial Metabolite Center website that's being created. Really the goal of this particular workshop and really the goal of the Collaborative Microbial Metabolite Center is really illustrated by this particular image right here. Even though here we focus on human health, it could be any ecosystem. It could be the ocean ecosystem, it could be a plant ecosystem, it could be almost anything. And so the start of these particular series of these workshops and the tools that we're building is to better understand microbial metabolites. And it turns out that understanding microbial metabolites in ecosystems is quite difficult. And we're not the only ones that recognize this. The National Institute of Health in the United States recognized that this was also a challenge. But if you want to functionally understand microbiota in ecosystems, you essentially need to understand the metabolites that it produces and their activities. And so NIH created a call to build a knowledge base of microbial metabolites. And that's where we created this Collaborative Microbial Metabolite Center. Is there a question in the chat? Oh, Elena, you can answer that one. So the Collaborative Microbial Metabolite Center is essentially a knowledge base that we're creating. And that there are grants awarded around this that create knowledge about microbial metabolites and their activities. And that data needs to go into this knowledge base from those awardees. And then we curate, store, and provide knowledge enrichment and provide documentation, training, etc. So with this center, we're actually expanding this because this knowledge is being curated. We also have new tools that we're developing, and one of those tools is what you're going to learn about today. But we're also reaching out to the worldwide community to capture this knowledge. And again, the tool that you're learning about was really came about together with the worldwide community. So the tool specifically that we're going to be learning about, so every workshop there will be different tools and different resources that we're going to give the workshop on. But today it's going to be on microbeMASST. And so think of an experiment that you might do. In this case, we have a mouse and then a mouse treated with antibiotics. But it could be a healthy disease state, it could be an intervention, observational study, and you have condition A and condition B. You can collect untargeted metabolomics on this. And so this is when you have both the precursor ions, so the intact ion of the molecules that are part of the sample. And then you also, in our case, we fragment all the ions that we see. So we get these MSMS signatures. But then you can do some sort of statistics on it, and maybe you prioritize some molecules that you see in the no antibiotic treatment and they disappear in the antibiotic treatment, and maybe some other ones appear. And so there's a variety of different methods that you might, or 1,001 methods that you can use to prioritize your signals that you're looking at. But it's very common that these signatures are not annotated. And if you want to answer the question is, who is able to produce these particular molecules? So in this case, we're looking at mice and then antibiotic treatment. And we want to know, well, is it really possibly the mouse or is it the microbiota that are producing it? And then if microbiota are able to produce it, what organisms are able to produce it? That's currently a question that's actually very hard to address. And this is one of the reasons why we developed microbeMASST. But microbeMASST has many other uses, but this is just an example. And so what you then do is you obtain the MSMS spectra, so the fragment spectra of the ion, and then we import that into microbeMASST. And I'll tell you in a second what microbeMASST really does. But this essentially allows you to hypothesize what organisms are potentially able to produce this MSMS spectra, and from that, we infer that it has the potential of making that particular molecule. Does anybody have questions so far?
I have maybe one question.
Yeah, absolutely. That's what we're here for.
Yes, so I wonder whether you hypothesize which bacteria are producing the metabolites given a certain genomic signature that was produced by the bacteria, and that's how you link it together, or how you do it?
No,
Maybe
and I'm going to show you how we're going to show you how we link it next.
Okay, sorry, yeah.
No, so it's in the absence of genomic signature, although in the future we would like to connect it to the genomic signatures as well, so that you can either come in via the genomes sequence, where you find certain gene clusters or genes that are present, and then you infer that that molecule is produced, but we actually do it based on mass spec alone. Okay, so microbeMASST is the following. It's basically when we created a library of organisms where we have LC-MS data, untargeted, of microbes grown in culture. And so now when you search with this MSMS spectra, you can search which organisms have the ability to potentially produce that molecule. We are pulling it back later on. So right now, later on in the workshop, you'll also see that if the structure is known and the MSMS is available, that you can search it as well. In the future, we are building that you can search based on microbe name. We have a crude way of doing that, and I will show you that in the workshop as well. But what we don't have yet is based on microbial biosynthetic gene cluster or genes that are present. We're planning on building that in the future. Right now, what we're linking is untargeted metabolomics data of microbes grown in culture, and they don't have an accompanying sequencing data. In the future, when we build microbeMASST 2.0, then we will expand that we directly link to whole genome data, or 16S, 18S, or draft genome data. And so that will be the future, but that's not there yet. Now, if you have data yourself, where you have LC-MS untargeted metabolomics data, and you have genomic data, get in contact with us, because that will help to build microbeMASST 2.0. Okay, building microbeMASST. So in where the data is stored, so all the untargeted metabolomics data is stored in the GMPS ecosystem. The redo format is our metadata format that we used. Giselle, you have a question?
Yes, Peter, but it is from the past slide. With regards with requesting data from whole genome data and 16S and 18S.
Yep.
When we upload the data, it was a link to the NCBI data. Do you have the means to couple? I mean, to intertwine this to data, or do we have to upload it again? How is it going to be?
So okay. So there's many different questions. So indeed, there are files that we've connected to NCBI data, but NCBI taxonomy is not phylogeny. So a lot of things that a lot of organisms that people are looking at don't have an NCBI taxonomy. So in other words, in that case, it's often not directly linked to NCBI taxonomy or to a sequence. Now, if the sequence that is deposited within microbeMASST is to the species level, then we can link it to, or the strain level, then we can link it to the genomic information if available. But that was only a small portion of the entire MAST repository or microMASST repository. Does that make sense? So the next step will indeed be linking, let's say, SRA accession number to, or even the 16S to the specific organisms that are present within the resource.
Okay, Peter, perhaps the question is, do we have to contact you again in order to link our data? Our data is based on 2 particular species, so.
It won't hurt, because microMASST 2.0 will maybe emerge in the next 2 to 3 years, and so beginning that process. And if you want to participate, yeah, it wouldn't hurt to contact us again on that.
Okay, and the NMR spectra, how are you going to be linked this particular?
So hold on, in terms of structure linking to MSMS?
Yes.
That will be covered in the next workshop, because we've created a specific upload portal to capture structures, genetic information, spectra, etc. But that's a part of the next workshop.
Okay. Okay.
So we have a complete interface that we're building for this to specifically address that those connections.
Okay. Well, thanks.
Yep. Okay, so building microMASST. So the GMPS is where the data is stored, the raw LCMS or metabolomics data. Redo is the metadata template that's associated with it, so that we can control the vocabularies very nicely. And once you have that, one of the things that I did is I reached out on social media to see if people had LCMS data of microbes that were cultured. And so we had quite a number of people that responded to us. So about 25 different research groups, 70 scientists from 15 different countries. And many of the people that contributed are part of this workshop today. And that created 100 million MSMS spectra associated with bacteria, fungi, human cells, and then also some QC and blanks. So when you look at the taxonomic tree, there's about 60,000 unique files that we have. They created about 2,300 unique taxonomic names. Those were blasted, resulted in 1,800 lineages. So filling in the taxonomic tree, about 2,800 nodes. Does anybody have questions about this? And if you do, Simone can answer, because he did all this work. All right. So we have sort of a distribution. This is the distribution we currently have. Hopefully over the next few years, we will expand this. Right now, we have about 60,000 LCMS files that we will be searching today in the workshop. My hope is, as the community starts to expand, that this goes to hundreds of thousands different LCMS files of cultured microbes as this resource continues to grow. What we then build the interface so that you can search with any MSMS spectra that you have prioritized or are interested in against these indexed spectra or files that we index very nicely. Now, of course, one of of course, we're dependent on data that's available by the community. So if you want to look if your favorite organism is represented in this data set, you can click on that Google spreadsheet. And you can start to see which organisms are present. If your favorite organisms are not present, then I would encourage you to get untargeted metabolomics data and then reach out to us so that we can begin to add this in the next generation. when we go to microBMASST, and we'll get to the hands-on portion in a little bit, but this is what the interface looks like. It's pretty straightforward. So we have an option to represent the MSMS vector as a USI, or Universal Spectrum Identifier. And I will show you how to get some of these Universal Spectrum Identifiers. Or you can enter it manually. Which means you need to have a table of all the fragment ions and their relative intensities, as well as the precursor M/Z. And you're going to be doing it hands-on today, both with an example of USI, as well as manual entry of the data. Once you perform a search, you have to, of course, specify your search criteria. And we can have a discussion what's a good search criteria, what's not. You can decide. And then you can hit search. If you have a USI, you search by USI. And then if you search by spectrum peaks, you hit the spectrum search by spectrum peaks. Now, microBMASST is only possible due to the collaboration with a large number of people. I'm not going to call them out individually, except for the first three, Simone, Robin, and Annelies. Because of them, a lot of this work was done. The interface was built, and then also Ming Wang at the end. But lots of people contributed to the knowledge and the information that's embedded within microBMASST. Does anybody have questions at this point?
Me again, sorry.
Yep.
From what I saw on the web page, you can, and what you have explained is that you have one MS spectra, and then you put it into microBMASST, and you get all the bacteria that are associated to that metabolite. I think we already did this question before with you, but I just wonder whether in the future, can you input actually a bacteria, and from this bacteria you get all the metabolites that this bacteria is associated with, like the other way around?
So basically, you're asking for batch MSMS processing. Oh, asking what metabolites are in the bacteria.
Exactly, are kind of interacting with the bacteria, or what the bacteria is producing, or so on. If I have actually
so the way
an organism of interest, and I go directly to that organism, and I get the profile of that organism.
Yeah. So the way I would do that is I would take the lists that I just showed you, the Google sheet with all the microbes and all the data files. I would grab your organism that you're interested in. So you can just do a search in the Google sheet.
Okay. Yeah.
Search, and then take those files and do molecular networking with it. Then you can find all the metabolites that are annotated through the GMPS ecosystem. So last month's workshop was on molecular networking, which you participated in as well.
Yeah.
And so that's the process that I would take at the moment. We don't have an easy automated way to address this, but on the other hand, it won't take that long, because you have all the I mean, you basically have all the MSV files, you have the file names, and so you can use that to create a molecular networking job.
Yeah, that's fair. Thank you.
Yep. Okay. Any other questions? All right. So we're going to go to the hands-on workshop. If you don't have access to the materials yet, you can go to tinyurl.com/CMMC-WS2. Maybe if Helena or Simone can paste it real quick in the chat for people, that'd be great. And I will wait 1 minute so that people can get access to it. And all the materials, including the presentation that I just showed you, are all linked. So this was the presentation that I just showed you. These are the presentations that I'm going to, or this is the presentation that I'm going to cover now. So all the material is available and accessible to you. All righty. Let me know if I need to slow down or go faster. Actually, slow down is what's sometimes needed. All right. So the first thing that we're going to do for the microMASST workshop, the hands-on portion, is doing manual MSMS entry. And so we've all seen papers where they don't make the raw data available. And here's an example of 2 papers where they have metabolites that are interesting microbiome-derived metabolites, but they don't have the raw data available, and that is the norm at the moment. So hopefully when some of you review papers and you find that they have untargeted metabolomics or metabolomics data, encourage them to upload that data publicly, whether that is in GMPS Massive or Metabolites, Metabolomic Workbench. There's also some other repositories, but the reason why people didn't make data available for a long time in metabolomics is because it's nobody saw the value in it. There's this general paradigm that you cannot reuse metabolomics data. It was taught for decades in analytical chemistry classes. We need to move away from that paradigm and actually start to realize this data is incredibly valuable. And so what we're going to do here is we're going to basically take a spectrum like this, and we're going to search it using microBMASST. And the fundamental question to really ask here is what organisms are potentially capable of producing this particular molecule? And so this is an MSMS spectrum that was from the supporting information. Here's another one that was a part of the supporting information. And again, the raw data is not available. And so this is on the bottom is the MSMS spectra of commendamide. And so what I've done in the past for papers who only have the image in the supporting information or in the paper, if you're lucky, they provide you some high resolution annotations of the ions that are present, of the fragmentation data. If not, it's okay to do low resolution. You just have to adjust your settings in microBMASST later on to account for that. But I've literally gone with a ruler, and I would have measured sometimes, or you can, if they gave percent intensities, you can estimate your percentages over here. And so I've sat there with rulers to just simply measure all the most abundant ions that are present and then created a table. And so the way I would have created the table is said, okay, here's 76.04, relative intensity is 100. If you had some measurement, maybe it was 10 millimeters or 11 millimeters, could have been fine as well. And you do that essentially for each ion that you see. And so then you get a table of M over Z and the intensity. And we know the precursor mass of this particular molecule is 330.264. So that's usually given in a paper. Does anybody have questions how to generate this? Great. All righty. Then we're going to go to the next step. So at this point, we're going to go to microBMASST. So if you had to open up this presentation, you can just click directly on the link. If not, if Helena or Simona or anybody else who's on can quickly copy and paste it and put it in the chat so that other people can follow me. But when you click on this window will show up. Is there anybody who wants to follow me that wasn't able to get to this microMASST link?
Sorry,
Great.
Peter, there is a question in the chat. Maybe you can go back to the previous slides. They are
Yep.
asking how to generate this. Yeah.
Okay. So there's so there's different ways. One, you can sort of crudely estimate, in this case, okay, here we have a peak at 76.038 and it's at 100. And so you literally have one column in the table that is 76.04, which is this ion right here. And then the intensity is at 100. If you are reading in a paper, often they don't have these intensities, they just have this spectrum. You can literally grab a ruler. I don't have one in my office right now, but you can literally grab a ruler and measure the length of this on the sheet of paper that you have, and then add that as the relative intensity. So, and then you go to the next one. Well, this one is maybe about 50%. And so I would now say 312.25 and then 50. And you do that for all the most intense ions that you see. If the paper did not add the mass to it, you can try to estimate it, but the error will be really high, which is okay. And you can still search with them, but just need to account for it. Does that help? So, and part of it is because, particularly if you look at, well, new literature doesn't always provide the raw data, but the old literature, all spectra are just in supporting information or in the paper. And so you have nothing else but to go and try to translate this into this sort of relative intensity. Because usually when you reach out to people that, let's say they published 30 years ago, they're not going to have the raw data anymore. They will not be able to recover it. In fact, if you reach the people that did the work 2 years ago, they will not be able to find the raw data anymore. It's really a bit challenging when it doesn't enter the public domain, but this gives you an option to still search. There are some programs that people have developed that can take an image like this and translate it. I've personally not used them, so I don't know how effective they are yet. So that's something to look into. Giselle, you have a question? Feel free to just interrupt and ask the question.
Yes, how do you select the noise level? I mean, there are multiple peaks there.
So that's a great question. That's a general question in mass spec. So for me, when I look, the noise sits right here. It probably sits in the 1 or 0.5% in this case. But in some cases, it sits at 40%, and you need to be the judge of what you feel like is noise or not. Mass spec always has noisy spectra, and it always has good spectra, and you just need to be a judge. If I look at these spectra around the sort of 75 to 150 range, if you go above the sort of 2.5%, none of those are noisy. So I would basically pick as many peaks that I can annotate here and translate it to the table. These two are obvious. This one I would probably include if I had an annotation, but I would probably not include if I did not have an annotation in terms of the mass. And the reason for this is even when you do a cosine correlation, which is how we compare spectra, this is such a minor contributing compared to the big ions that you see here, that not including that is probably not going to change the answer that you're going to get. Does that make sense?
Yes, thank you.
So, okay, so now we're here. And we're first going to focus on manual entry. And so what we're going to do is we're going to take the precursor mass. We're going to add that to the precursor mass. So you can simply copy and paste it. And then, so precursor, we're going to ignore the spectrum identifier. So the first thing that you're going to do is, well, you can either add the precursor mass first or copy this table on the right. So, whoops. Copy the table on the right and then paste it into spectrum peaks. And I'm going to go do it myself real quick. Gonna open up microbeMASST. And this has never been tested with this many people, so there may be some bandwidth issues. We'll find out soon. So we're going to paste in the precursor mass. We're going to copy and paste the table. Okay, I'm personally going to fix some of these things. Not sure if that's needed, if there's some shifts. You can set your cosine threshold. So you have a couple of options that you can change. You can change your parent mass tolerance. So that's the filtering that happens for all the spectra that you're going to consider. It would have to be within 330 Dalton plus or minus 0.05 Dalton. You can increase or decrease that. Obviously, if you go from a spectra where you're not sure about the error tolerance, you can increase this quite a bit. If it's a low resolution instrument, you're going to have to increase this a lot. Then we have fragment tolerance. Again, we set it in this case because we're looking at high resolution data mostly. Set it to 0.05. Cosine threshold. I often like to use 0.6. And then often the minimum match peaks. I personally like to set this to 5 or 6. If you, so yeah, so I think these are some important settings. If you set this to 3 or 2, there's going to be a lot of spectra that match that are incorrect. When you set this to 5 or 6, the false discovery rates tend to be pretty good. If you want to learn about false discovery rates, there's a paper on false discovery rate of spectral matching that we've published on with the Booker Lab. Maybe if somebody can find it and paste it in the chat, that would be useful. Okay, now you have your settings. And now you have to search by spectrum peaks. And then,
I'm sorry, I have a question.
Yes.
Based on like, how do you choose the cosine threshold?
So the cosine threshold, yeah. So what we've seen in the past, and so we do this partially based on empirical observations. When you have a cosine of 0.6 to 0.7, we tend to have about a 1% false discovery rate. And for us, that is acceptable. If you want less false discovery rate, you want to increase that to, let's say, 0.9. But one of the challenges is we're searching public data at this point. And so if you were to have done a really well-controlled experiment where you have run all the cultures on the same instrument, and then your experiment was run on the same instrument, that MSMS should look almost identical. In that case, I would raise the cosine threshold. But if, whoops, that's not the one that I wanted to show. So if you did not control that experiment very well, or if you're searching public data like we are now, there's often some variation in these intensities, and that's going to affect the cosine correlation. It still will require that the same peaks are there. It's just that there's some variation there. And so that's based on what we've seen in the past, and based on the paper. I don't know if somebody pasted it yet in the chat, but based on what we learned from the paper with Sebastian Booker on false discovery rate estimation, usually a cosine of 0.6 gives you around a 1% false discovery rate, and that's one of the main reasons why we commonly use it. Okay, so when you hit search microMASST, there was some thinking, and then you can scroll down here.
Peter, you are not sharing anymore.
Oh,
your screen.
I'm sorry. Okay. So when you hit microMASST, there was like a little blue thinker. It takes a few seconds to get the results. And then when you scroll, you can start to see the tree. So this is the tree of all the organisms in this particular microMASST repository that we now linked with the metadata that have this MSMS spectra within your search criteria. So if you're more strict, you will find fewer. If you relax your search criteria, you're going to find more. And so it's really you have to be the judge of what you feel like is acceptable results. But let me carry you through what this gives for information. First of all, this MSMS spectra is only observed in prokaryotes. So in other words, it's not found in fungi. It's not found in cultured human cell lines. It's only found in prokaryotes. It's only found in bacteria. And then when you look at the organisms, Bacteroides, cyanobacteria, Firmicutes, and Proteobacterium. And that is at the phylum level. And when you hover here, it shows that there's yellow, and sometimes there's some blue. And if you really squint carefully, you can see that there's a blue sliver in there. And that's basically the pie chart showing what percentage of the Bacteroides have this particular MSMS spectrum. So in this case, it's 13 out of the 1,492 samples where we have LC-MS data that have an MSMS match to the Bacteroides. We can expand this a little bit. Now you can start to see, okay, 3 out of the 1,342 Bacteroidida class have this one. But if you now look at Cytophagia, it's 1 out of the 3 samples, and Flavobacteria, it's 9 out of the 75. We can further expand this. You can expand it whichever way you want. You can further expand sort of what's in the neighborhood if you want. And there's some interpretation that's really important. So first of all, you don't see it in the blanks, eukaryotes, and QCs. So that's encouraging. But let's say we find it in cyanobacteria. We find it in 4 out of the 4,600 or so cyanobacteria that we have. Okay, that's great. But none of the ones where we have order information. And so we only have order information for these. It basically means that probably the NCBI taxonomy that was defined for many of these organisms, for many of these samples, were only at the phylum level and not down to order or species level. In other cases, we do get all the way to species level. And so here, the Ludibacter flavus, we see 1 out of 1 samples have that MSMS. Here we see 2 out of the 2 at the genus level. If you look at Flavobacteriaceae, 9 out of the 63. And so it gives you a hint, sort of a guideline of which organisms are potentially able to make these particular molecules. And so if you now go, if you think about the Commendamide, those original papers were really beautiful, but it's clear to me that that molecule is quite commonly produced by a lot of different microbes. Does anybody have questions so far?
I have maybe 2 questions.
Yep.
Sorry, again, me.
No, by all means. This is what we're here for. This is why this is a nice hands-on workshop.
Yes. So at some points when you, for instance, in the bacteroidetes side, if you go at the end to the right, there is the Bacteroides fragilis, which is at strain level.
Correct.
1 sampling out of 7.
Correct.
That means that it might be certain, I don't know, strain differences across the same bacteria. That's why it's not produced in the other 6, but it's produced in 1, or it's just because maybe people from different labs uploaded this information over there and just didn't match the metabolites that they found.
So that's a great question. So there's yeah. So indeed, so here you have lots of several strains, and one interpretation could be, and so that's those other strains are not able to produce it. The other interpretation is that maybe it's only produced under one condition, and this particular strain has been cultured in many different conditions, and that data is uploaded. So what you want to do is probably go back to the raw data, and I will show you how we can get to the raw data to see if this was done in one experiment and they're cultured in the same condition. And you can go back to the raw data and see, okay, do they have those particular peaks or not? The other thing could be that maybe somebody uploaded the specific extraction conditions that the other ones don't have. And so what this is doing, it allows you to initially build a hypothesis, and then you need to go back and ask those specific questions.
Yeah. Okay.
Does that make sense?
That makes sense. Thank you. Thank you, Peter.
Yeah, that's the challenge. It's a slightly different interpretation than if you have a genome. It's a genome. There's either presence or absence of genes. Whereas here, there are lots of different culture conditions. So let's say the right precursors weren't available in the culture media, you're not going to see the molecules. If the right culture and conditions exist, then you're going to see those molecules. And so you have to keep that in mind. But at the same time, it's for the first time that we can start to create this sort of taxonomic assessment of candidate molecules. But you do have to interpret it with that in mind. Okay, so any other questions?
Can I ask something quickly?
Yes, absolutely.
Yeah, this is really interesting so far. Thank you. So I was just quickly, I know this is micro focus, but I was wondering if other taxa would expand as well. Not taxa, phyla, sorry.
Yep.
Cool.
So we are currently developing plant mass where we have taxa for plants that are being mapped. However, we are over the summer. Hopefully, we're going to expand this to pretty much all organisms. And so you will start to see the relationships. And there it's going to be really interesting because let's say you find it in humans, mice, and other animals, and you find lots of matches to microbes, then you can say, well, likely that molecule that's found in those human samples or mouse samples or other animal samples probably originate from microbes.
Very cool. Thank you.
Yep. That will be the next phase of this effort. Okay, so then we also have library matches. You can click on that. And what library matches does is searches against all publicly accessible reference spectra of molecules. So it has HMDB in it, it has MONA in it, it has ReSpect, about 30 to 40 different spectral libraries that are publicly accessible are part of this. In addition, there's a suspect library that we have created that is a part of this. And there's a preprint on this if you want to read more what the suspect library really is about. But it means it gives you all possible annotations that are available within your search criteria. And I need to re-emphasize this. And there's a couple of things to look at. So first of all, it will give you the GMPS library accession number. You can click on it and you can find the information. Gives you the universal spectrum identifier. It gives you the charge and then how similar it is based on the cosine and then number of peaks that match. And to me, for me, when I interpret quality of matches at a quick glance, what you want to see is you want to see the cosine high and ideally the number of peaks high. If it's above 5 or 6, it generally is a pretty decent match. And then it has the compound name. And so this is a putative hydroxyhexadecanol glycine, which is the sort of more scientific name for commendamide. Then below it, we have suspect library. And it basically tells you that it's very similar to commendamide again, et cetera, except there are some substitutions. Does anybody have questions on this? So what you sometimes see if you search, let's say, some of the new bile acids that are made by microbes, one of the things that we'll do later on, you might get 10 matches, 50 matches, or you may get 0 sometimes if it's an unknown molecule. All right. Then the other thing that we have is you get data set matches. And so what is really nice about this one is, so here we have a match to E. coli nissile, or we have the match to bacteria theta micron. You can actually open this file and it will actually bring you to the raw spectral information. And so it takes a second to load. If it's never been downloaded before, we move it from the main GMPS web server. We have to FTP it over. Since it's one file, it may take up to a minute. But basically, it uploads the USI and it will take a second. The file download conversion is being done. And then it will draw or basically create a plot of the LCMS run that we're looking at. So this is the LCMS run. We have the retention time. We have the M/Z value here. And then each of the blue dots or stars is where there's an MSMS spectrum. Now we can filter for the specific M over Z value, which was, I know, 330.264. Oops. 264. I'm going to filter based on ppm. So I want it within 10 ppm. And once you do that, it will filter it and it will plot the XIC, so selected ion chromatogram. And so you can inspect it. And then here, the red stars now is where the MS2 acquisition was. And you can click on one of those. And you can start to look at if those key ions that you entered are present. So 76.04 was present, which is a part of your table, etc. So you can manually inspect the data associated in the actual file that was deposited in the public domain. Does anybody have questions? All right. Then we also have taxa matches.
Sorry, maybe you said that, but can you download the raw file?
Yeah, all this can be downloaded. Usually it's easier to go to the actual file itself. And so if you go to GMPS Massive, this is the accession number, and then this is the file name. And so if you go to gmps.ucsd.edu, you can go to the data sets, and then you can download that specific file. If you want to use it to do molecular networking of this file, you can also just simply hit click and perform molecular networking. Does that help?
Yeah, thank you.
Okay, great. All right, so then we have taxa matches. You can see all the information associated with it. It's basically here this is how we represent this graphically. Here we represent that as in a table format. And again, all of these you can download as an Excel CSV, or you can copy and paste it. And then the parameters basically shows you minimum, basically your parameters that you used for searching. So that's that information is retained. Alrighty, yeah, go ahead.
I'm curious, what's the difference between the library match, database match, and the text match? The difference.
So the taxa match are so library matches. So if you have a reference library of known molecules, that's the library.
Okay.
Does that make sense?
Yeah, yes.
So that is nice if you get a library match. If you're searching some spectra and you get a library match, it's really nice because you also get structural information. Whereas if you don't get a library match, then it's an unknown. It's or at least there's no reference spectra in the public domain that has an annotation, and so it becomes a little bit harder to interpret. Whereas if you have taxa matches, the annotation is based on the taxonomic tree.
Okay.
And so there won't be a structure necessarily attached to it if you have a taxa match. And so you can get taxa matches and not a library match, but sometimes it also happens that you get a library match but no taxa match. And that basically tells us that our reference data that we have for microbes doesn't have that MSMS spectra in it. even though the molecule may be known. So those scenarios happen.
Okay.
Does that make sense?
Yes, thank you.
Great. Okay. Now I need to go back to my presentation. So does anybody have questions about the manual entry of MSMS factory in microBAST? Great. So the next one will be using a universal spectrum identifier. And if you want to learn what a universal spectrum identifier is, you can go to this preprint. We deposited in 2020. We still haven't created the revisions. It's not that important to us, but eventually we will have a paper out on this. But essentially, the information that's associated is really all the spectral information. It's a unique identifier, and that's really important. So this tells us that this universal spectrum identifier is a mass spec. We look at the GMPS repository, and then the GMPS library accession in this case with the unique identifier. But different repositories have different identifiers. So if you have MassBank, for example, which is another structural library associated with the metabolomics community, there you identify MassBank as the resource and then accession number. If you go to the massive GMPS repository spectrum, then you have the accession number of the project itself, the file name. This one, they have a bit of a long file name, and then the scan number. And so this is how the universal spectrum identifier is identified, and then you can put that into your microBAST query. And so if you go back to the microBAST webpage, we have a couple of examples that we're going to just use today. But I want you to click on the glutamate cholic acid, and then you'll see that this spectrum identifier shows up. Is there anybody who wanted to follow me that was not able to get here? So glutamate cholic acid are new microbial bile acids that have been discovered, and this is just one example of it. What we're now going to do is we're going to find out which organisms have the ability to make that particular molecule. And so once you click on the glutamate cholic acid, you then hit search by microBAST by USI. And when you do this, this window will show up. And what is nice to see is that if you look at these organisms that show up, you'll start to see that most of these are really animal or human microbiota. We have lots of strains from soil. We have lots of strains from ocean environment. We also have fungi, but only bacteria show up. And they all belong to human and animal microbiota. We can explore and discuss a little bit. Let me go to microBAST myself. All right. So again, now we can go to library matches. You can start to see a couple of really interesting things. First of all, we have, so that we only show 4 matches at a time. We have 9 of these, so there's almost 40 different matches that we see. Here, glutamate, cholic acid, suspect related to taurocholic acid, but with a delta mass of 22.039. The difference between taurocholic acid and glutamate cholic acid is 22.039. Again, we get a match to glutamate cholic acid. And we can keep going down this line a little bit. Glutamate, conjugated cholic acid. So these are all different annotations. But as you go further down and your cosine gets worse and worse, you can start to see now we have a glutamate, but this is connected to gamma muricholic acid. And so one of the things that will happen is when you search these MSMS spectra, it will often match to isomers. So that's something to keep in mind, that when you search with MSMS spectra, sometimes you can't differentiate isomers. At least not at the search level within the search criteria that we specified. Does anybody have questions about this? All right. So then we can explore. You can show up to the phylum level if that's what is of interest. You can, oops, going the other way. Genus level or class level, genus level. And then there are a couple of other things that we have here. So let me go to the maximum level. So here we also have a filter of minimum matches. So oftentimes, if you just get a single match, it may be less reliable than something that's matched 10 times or 3 times or 4 times. And so you can increase this. Whoops. And you can filter for things where you at least have, let's say, 3 matches. And so you can start to see that your tree gets much smaller, but you can increase your confidence that this is really commonly observed in those particular organisms. So that's a useful thing to do. You can also size for ratio. So right now, you can also size based on number of matches that you have. You can size it based on the number of samples that are there, and then you can hit scale. So in other words, here we have much fewer samples in the Bifidio examples than, let's say, if we look at E. coli Nissal 1917. And so that's sometimes useful to interpret. Here's number of matches. Here's the ratio. Yeah, go ahead.
Yes, so because here in Escherichia coli, you have 3 kind of matches, but out of 167 available samples, but for instance, in Bifidobacterium, you have 1 match, but out of 5 or 6. So would it be also good to represent them as percentage, this minimum matches thing, just from the available sources that you have here in the database?
Yeah, so we have not created the ability to visualize it by percentage, but if you hover over it, it shows the occurrence.
Okay.
And that is the percentage.
Yeah.
Right. So we do have the ability to scale by ratio. So in other words, if let me go to 1. So as the ratio gets larger, the node size gets larger.
Yeah, thank you.
So that's essentially what you're asking for, except presented in a slightly different way.
Yeah, exactly. Thank you.
All right. So this is really getting close to the end of the sort of workshop at this point. At this point, what I would love you to do is just navigate through some of these examples that we have. You can ask the following questions to yourself, and if you struggle with answering these questions, so for example, lovastatin, if you click on this, is this made by humans, fungi, bacteria, or archaea? Selenosporamide, is this made by human or animal microbiomes? Yersinia, this was originally isolated from Yersinia pestis. Do other organisms make it? Microferrioxamine. I know the human microbiome has lots of receptors for ferrioxamine, but can human microbiota make it? And then citrulline deoxycholic acid, who makes this molecule? So maybe in the next 5 or 10 minutes, this is really the end of the workshop. The rest of this will be discussion. If you have questions, etc. I will stay on for another 50 minutes. What I may do is show you an example of how we've used it or started to use it. That may be good to see as well. Let me go through this. So one of the experiments that we were really interested in was to try to understand how microbiota influence the chemistry of the entire host of the entire organism. And so we had germ-free mice colonized with a normal, healthy microbial community, which essentially gave us about 1.2 million or almost 2 million MSMS spectra, but 30,000 unique MSMS spectra. We then searched this with microBMASST, so we had about 10,000 spectra that was different between the germ-free mice and colonized. And then we searched those 10,000 spectra with microBMASST, and about 3,200 of those MSMS spectra were found with microBMASST. We then removed all the MSMS found in human cell lines, which were about 800. And then we validated against another study where we had mice that were treated with antibiotics. And 512 of those were found to be associated with mice that were not treated with antibiotics that disappeared. And so when you then search those 512 MSMS spectra, you can start to assess what organisms are able to produce these MSMS spectra, or these same molecules from which these MSMS spectra originate. And then we searched larger and broader to see if those were found in humans. And then we could start to find where these molecules are. And so this is one example of how you can begin to think about using this microBMASST resource. Anyway, as you go through these examples, this is the end of the workshop. As you go through these examples, just feel free to ask us questions. Questions about the results, questions about interpretation. Now is the time.
Hello, may I ask, please?
Yeah, of course.
Great. So very useful. I'll try to go through more. I'd like to ask, when you run more than one MSMS features, does it help the one the other on the final prediction in a way that Zodiac, let's say, help in the final prediction, so they don't, as we assume that they would come from the same experiment, if the one is from human microbiome and the other is from fungi, does it help for the final prediction to reduce the chances and say, okay, probably it is that candidate because the other 5 features that you have run, they're also from the same direction?
So let me rephrase it to see if I understand your question. And Helena or anybody else, if they can chime in, that would be great as well. But what I'm trying to understand is that when you do microBMASST searches with multiple spectra, you get results associated with the taxonomy for each of the spectra, correct? And so that gives you a tree. And so now you have 5 different trees. And so the question that I think you have is, if you have multiple matches to one part of the phylogenetic tree in those 5 examples that you search with, that then increases your confidence that that organism is responsible for making it?
Yes, like, okay, my cosine score might be, let's say, 0.8, but because in the other 5 features that I have run at the same time, they have the same candidate, instead of 0.8, I evaluate as 0.9 or something like that. Does it reconsider based on the other results?
No, so it does not consider based on the other results. However, you can create a creative way of interpreting it. So I think it's a great question. We've not developed a strategy, and hopefully you're welcome to create it yourself. But we've not really developed a strategy that if you search more than one spectra, that it sort of rescores the metric, so that you can say, well, it's more likely that it's going to be this organism and not that organism. We don't have such scoring metric. I think it's a great idea. Honestly, we just don't have that yet. We're very early in being able to do this. So this is the capabilities that you saw today is really the first step towards being able to develop those kind of creative tools with the community. I think it's a great comment.
Excuse me.
Yeah.
I'm curious about the intensity, the mass data intensity. When the wrong method, such as the temperature, the voltage is different, the mass method different, whether intensity change different.
So I mean, so this goes beyond just temperature changes and those kind of things. So temperatures, so intensities do change depending on the instrument settings.
Yeah, yes.
So that's expected, that you get some variation in the spectral information. Now, your search criteria dictates what you're willing to accept as a good match or not a good match. And so if the data is collected under such different conditions, then your MSMS spectra, you're just not going to find a match.
Okay.
So that's it. That's why I encourage to the community, upload as much data as possible, because the more instrument condition, the more different instruments we have, the more coverage we're going to get. my hope is that this is going to be 10 million LC-MS runs in the next 5 years. It will, that sort of scenario, and that each organism is covered by 5,000 different LC-MS runs, so that it's much easier to bridge that gap. Now, we do some tricks when we do scoring matching within the GMPS ecosystem to improve as many matches as we can. One, we filter for the top 6 ions usually for every 50 Daltons. And so that eliminates some of the noise. The other thing is we apply a square root. And that decreases the high intensity ions a little bit and increases the low intensity ions a little bit. And so if there is some variation, it will remove some of that variation. It won't eliminate it, but it will move some of it. But again, I encourage people to upload as many data files as they can for a given organism.
Okay, thank you.
Yep, you're welcome.
My second question is, can we use this method to find new compound or some novel compound?
Yes. So the compound can be completely unknown. And what it will do is it allows you to find taxonomically who is likely able to make it. And so let's say you do a natural product research project and you find that you have a really interesting compound. It looks to give good activity in the fraction that you're looking at, but it produces very little. Well, sometimes what people can do is search with the MS spectra and find other organisms that can make it, and maybe one of those other organisms produces a large amount of it. Well, then you want to culture that organism instead of the one that you're working with. So that is one application for sure.
Okay. Yes, good. Thank you. My third question was, when we get the metabolites from the bacteria, from the microbial, we always extract the impurity, extract some compound from the media. How can we make sure we get the data, we get the metabolites, it's from the bacteria, not from the media?
Yeah, this is a really good question. And so one, if you look at the results, let me show you. When you look at the results, and we expand the tree, and we can do the full tree if you wish. It'll take a second. Actually, that was probably a better view.
We are not seeing your screen.
Oh, okay. Let me share my screen again. Thank you for letting me know. Give me 1 second. Sorry, I have many windows open. All right. So the first approximation can be seen in this window. So this was the CMMC. So there are QCs and blanks. A lot of the blanks actually contain background. So in other words, where the microbe has not been added.
Okay.
So that's one thing that I would first look for. The one challenge, however, is with metabolomics, there's commonly that you have carryover. So some blanks that are added here do have carryover. So that's just something to pay attention to. The second way, what I would do is, since we have the data set matches, I would actually go to the original public repository, and hopefully the people who uploaded the data have all the QCs, all the blanks and background media, and I would compare it, your particular compounds that you have found, to the blanks. What we're going to do in the future in microBMASST 2.0 is we're going to try to link every background to every file, which was not possible to do at the moment because realistically, untargeted metabolomics data deposition is a mess. It's not well organized. Often blanks or controls are missing. And so we could only work with what we had available to us as a community. But those are the two options. Check here real quick. Sometimes you'll get a blank that matches. It doesn't mean it's guaranteed that it's really in the blank. It just means that you need to be extra careful with interpreting it. I think if you go through some of the other examples that I'm pinpointing out here, you'll start to see that some of the blanks and QC controls have that compound in it. But if you start to think about it, it does not make sense. The other thing, yeah, does that help?
Yeah, yes, good, thank you.
Great. Any other questions? So I would really go through these questions. Can you answer them? And if not, then let's have a discussion. Patrick Bercio, is it possible to search for a species or genus and find a unique spectra for this taxonomic clade? So, Patrick, the way I would do it, so it's a deconvoluted way of answering your question. I would go to that big list that we have of 60,000 LCMS files of microbes in culture, find which species or genus you're interested in. I would then do molecular networking, and then I would take those spectra that are not found in the blanks and do microBMASST on it. Hope that helps.
Yes, thank you very much.
You're welcome. I mean, you're in Bremen. That's the mass spec capital of the world. Both Thermo and Bruker headquarters live there.
Yeah, Bruker are our neighbors actually. They're just across the road.
Yeah. Any other questions? Well, we're gonna stay on. So, yeah, there was a question.
Yes, it's a very basic one. Since you have processed so many samples or microbial samples for LCMS, is there like a recommendation on how to get the best molecular breadth in terms of sample processing? So let's say if I have like a bunch of microbial strains I want to process, if there's a resource that basically yeah, because I know, I mean, the breadth of protocols and everything is broad, but
yeah, I think this is a really hard question. Because there's no simple answer. That's
Yeah.
the unfortunate part.
Yeah.
What we do in our laboratory, we've been consistently doing ethanol or methanol extractions.
Mm-hmm.
And that has given us a decent breadth. And so we would culture it, let's say, in 100 or 200 microliters if it's liquid. Or even on agar cultures, we grow a single colony and then take the single colony and extract that with typically either 95% ethanol or methanol. But if you're interested in less polar molecules, of course, you're going to have to do a different extraction. And then we run it in by LCMS. We tend to use a 15-minute gradient.
Mm-hmm.
I don't know, maybe, yeah, so it's a pretty standard chromatography method with acetonitrile that we use.
Mm-hmm.
And then for the instrument settings, we tend to, on a thermo instrument, the 30 collision energy. It's an arbitrary unit. In Bruker, we do a gradient ramping typically to get the MSMS spectra so that you get multiple sort of collision energies. And in terms of data dependent, we usually take a precursor scan and then we fragment the top 10, put it into an exclusion list. We leave it in the exclusion list typically for 1 minute to 2 minutes. So it's pretty standard settings that we tend to use. Now, if you have a molecule or molecule class that you're really interested in, but doesn't fragment very well, you're going to have to increase that energy.
Or the majority of extraction, basically the majority of sample processing in the database so far has been done along this. Let's say if one is interested in a lot more non-polar compounds, but the extraction. So what I mean is if I extract things that just haven't been extracted before, I guess one just has to hope that the database grows.
Yeah, one, you have to hope that the database grows, but for this is 60,000 LCMS runs that is done by the community.
Okay.
And so in one way it's a small number, but in a other way it's pretty representative of what the community has seen.
True.
So it may indeed be that if you have a specific MSMS spectra of a specific molecule that you don't see it. But one thing that people don't really appreciate, that even though if you do a non-polar extraction versus a polar extraction, generally about 60% of the molecules or 70% still overlap between the two. Okay. And so if you have an intermediate extraction protocol, then you're probably going to be 80% in both directions that still overlap. So it's pretty, I mean, it's a pretty decent coverage. It's not, you can never be perfect. But it's the best we can do at the moment.
No, but that's certainly, that answers my question.
Yeah.
Thank you.
I have 2 questions.
Yes.
So one question is if it's possible to produce this USI spectra, like if we have experimental spectra that we want to put in without doing the like a spectrum peaks search, like maybe we have many and there is a way of converting them into a USI in some way, so we don't have to put manually the masses and the intensity.
Yes. So my recommendation is to upload this into a repository.
Mm-hmm.
And then anything that's in a public repository of data files, you can create a USI with it. So that's pretty straightforward. There are ways that you can create a USI on your end. So I think that's described in the original paper, but the best strategy is really uploading it to a repository and then creating USIs with that.
Okay. Okay, thanks. And the second question is
before you go, are you familiar with how to upload to public repositories?
I mean, I did it for these projects. So I just have
okay.
to refresh the memory.
Yeah, we also have a drag and drop menu. So if you go to, there's a paper in Nature Metabolism that we published maybe a year ago or 2 years ago. Maybe if somebody can post it in the chat. It gives you direct instructions, but if you have a few files that you want to upload, the drag and drop mechanism is probably the most efficient one.
Okay, good. Yeah, I will look into the link if someone sends it in the chat. Yeah, and then the other question is in this, I mean, in the future, you said you plan to include more the files from different organisms. Does that include also potentially like fecal samples from human?
Yes. Yes.
Good.
Yeah. So that will be a future workshop, but the whole idea is, so we're going to add human diseases, different tissues from humans. We're going to add interventional studies, so that when you search in MSMS spectra, you can learn, okay, it's found in these diseases and not these diseases. It's found in these interventions and not in those interventions. We'll do the same thing for animals, where we have antibiotic treated animals, monocolonized animals.
That's gonna be amazing.
Diet intervention, all of these will be added to this, and so that it makes it much easier to interpret. We already have an early version of this, but it's not quite ready yet. And in fact, if people have data sets in the public domain where they're very carefully controlled, let's say an animal study, human study, reach out to us. We would love to make this a part of this search engine. What we need to do is basically curate the metadata so that it's very clear which samples were a part of the intervention, which samples were controls, so that we can create essentially similar to a phylogenetic branch that they experiment and control. Monocolonized with organism X, Y, Z. Antibiotic treatment, X, Y, Z, so that we can make it easier to interpret the relationships of each of these spectra to all these different conditions.
Okay, so like just related to this question, I guess at the moment that question that there about salinosporamide, is this made by human or animal microbiome? It cannot be answered.
Okay.
Or can it or not?
Okay, let's go through that. Let's go. Why don't you share your screen?
Me?
Yeah, Helena, could you give her permission to share her screen?
Yeah, okay, you can share now.
Thanks. Share this. Yes, I just put in the salinosporamide, I believe.
Yep.
Yeah.
Okay. So, so is this made? So click on the tree matched. No, yep, right there. Match other, perfect. So you can at least build a hypothesis to this question, right? At this point. So is it made by humans or human microbiota? And so now what you see here is, one, you don't get any matches to human cell lines. So this favors that it's made by humans, right? Do you agree with that?
Yeah.
Okay. You also don't see it in fungi because none of the eukaryotes match. Okay, you only see prokaryotes. You only see matches against what organisms?
Bacteria.
Yep. Keep going.
Actinobacteria.
Yep.
Actinomycetia.
Yep.
Micromonosporales.
Yep.
Sporaceae.
Mm-hmm.
Salinispora. Salinispora.
Correct.
Yeah.
Now, the question I have for you, are these found in human microbiota, in human sequencing data sets?
I mean, if I can see that by this outcome, by these results.
So here you need to know a little bit of knowledge. You can go and you can do a Google search on Salinispora.
Yeah, I searched that is, I searched actually this specific compound.
Yep.
And it says that it comes from marine bacteria.
Correct. And so does this mean that this molecule is made by human microbiota?
No.
Correct. So this is so that's the answer to that question.
Okay.
But it's beautiful that it's just, you can see that it's made by Salinispora. So somebody who cares about natural product discovery, in this case, Salinispora makes a really potent anti-cancer agent that people care about. That is in phase 3 clinical trials. But if people care about natural product discovery, small molecules that have bioactivities, and they want to know what organisms can make this specific molecule, this is the way to answer it. And in this case, you could clearly state how it's not even made by microbiota. So if I were to see this in a human sample, it would probably be a person that was a part of a clinical trial.
Yeah, I got it. Thanks.
Yeah.
But what I wanted to go to is that potentially now, let's assume that I didn't have Google. That potentially, like whenever we will upload also the human data, like the fecal samples, for instance, or the feces from mice, then you would be already able to see that by the outcome of the search, because then I wouldn't see them in the human and in the animals, right?
So I think your question is interesting. So basically, if you do indeed have a human sample, you ran CMMC on it, and you want to see if it's potentially made by microbes. And let's say you were to get this match to Salinispora tropica. At that point, one interpretation could be, well, it's possibly indeed a microbial molecule that is present, or maybe the host is able to make it, or it's a part of diet.
Yeah.
And so we don't have the diet information yet, which will be a part of microbeMASST 2.0. We do have food masks, which is similar, where you search MSMS spectra. But if you just get this answer, you basically get a hypothesis, okay, this MSMS spectra, the molecule that generated that MSMS spectra is possibly made by microbes, possibly made by Selenospora tropica. However, later on, when you do sequencing, you find out, well, I don't have a Selenospora tropica in my sequencing, so it cannot be making it. And that's the challenge when you have an incomplete database. If we had every organism in the human microbiome cultured and people had subjected this to LC-MS analysis, then it wouldn't be very ambiguous. But in this case, you can likely say my hypothesis will be that it's made by microbes because we do get matches against microbes. But in the end, Selenospora tropica only lives in the ocean, not in humans. Does that make sense?
Yeah, definitely. Well, thank you. And I guess we will contact you for contributing with all the fecal samples that we have from all our human studies.
Awesome. Thanks so much.
Yeah, I should stop. Sir.
Any other questions? One thing, Helena, could you paste the questionnaire in the chat? So we have a questionnaire. One of the goals of the questionnaire is to figure out what other types of workshops that you're interested in. We can organize even sequencing analysis workshops or in multi-omics integration or deep metabolomics inspection and how to prioritize what might be a microbial metabolite. Or any other recommendation that you might have or interest that you might have. Anyway, this is the workshop. So this is the end. Thanks so much. And if you have any other questions, let us know.